Good evening, everyone. In these times of virtual learning, where teachers and students have this only wireless technology to reach out to one another, webinars like such, given by some experts on various topics, can help to do the needful of reaching out to some more people at the same time at large. Good evening. I, Shonali Ray, on behalf of Department of Botany, Surendranath College, am privileged to welcome you all to this webinar this evening on techniques for estimating nucleic acids and determination of nucleolar frequency, to be delivered by Honorable Professor Sandip Mukhopadhyay, ex-head, Department of Botany, University of Calcutta. This is the first outreach program organized by our department under DBT-Star College Strengthening Scheme, Government of India. We have organized this keeping in mind the interest of students and hoping that the knowledge gained from this webinar lecture would help them in their curriculum. Now, before proceeding further, it's a humble request to all the participants to please kindly mute your microphones and keep your videos off. And if you have any queries or questions, please write it down in the chat box. All queries will be put up in the question answer session. So now I would request our respected principal, Sir Dr. Indranil Kaur, to kindly deliver welcome address and inaugurate this webinar. Over to you, Sir. Dr. Indranil Kaur.
Thank you, Shonali. Am I audible?
Yes, sir. Thank you, sir.
Good evening, everybody. It's a long innings of COVID-related pandemic. I should say it is a hyper pandemic. And this has created a lot of confusion in the regular day-to-day life of ours. But it has definitely produced some opportunities for us too. During this long innings of COVID-related lockdowns and others, we have learned to live with the online system of delivering our views and ideas and trying to cope up with this. And to my, I feel very nice that my departments, my college departments have also taken up this wonderful opportunity and started delivering their best and organizing various experts in different fields and bringing them closer to our students. And with their help, our students and faculties are getting enriched every day by the way of webinars and sharing knowledge with the experts in different areas. Today's webinar, definitely, it has the meaning, and nucleic acids are always really interesting for us. And every day we are finding newer opportunities in the discovering of various aspects of nucleic acids. Now today, it will be Dr. Professor Sandip Mukhopadhyay's turn to let us know about the latest techniques in understanding the nucleic acid in a much better way. I hope my faculty members and the students who are in the today's platform definitely will be enriched by the end of this program. With this hope, I welcome you all to this wonderful session of today, and expect by the end of this you will be enriched by a lot of with the help of Dr. Sandip Mukhopadhyay's lecture. Thank you very much.
Thank you, sir. I would now request Dr. Nilofer Khatun of Department of Botany to kindly give the honorable speaker his due introduction.
Thank you, Shonali. I hope I'm audible to you all.
Yes, you are. Thank you.
Good evening, everyone. It really gives me immense pleasure and privilege to introduce our speaker for today's webinar, Dr. Shondi Mukhopadhyay, retired professor and ex-head, Department of Botany, CU. Now, Professor Mukhopadhyay did his MSc in 1978 and completed his PhD in 1985 from CU. His research area includes plant biotechnology, molecular and cell biology, and cytogenetics. He did his postdoc research from Laval University, Quebec, Canada, and as well as from CU. He has been PhD mentor to both national and international students. He has many research papers and books published in international and national acclaimed journal with high impact. Also has couple of first reports to his credit. So without further ado, I would request Sir to please enlighten us with his lecture on techniques for estimating nucleic acid and determination of nucleolar frequency. Over to you, sir.
Thank you, Nilafar, for your kind introduction.
Thank you, sir.
So, good evening, everybody. My dear students, my dear colleagues, and other participants. First of all, I'd like to thank my students, particularly Shonali and Deeposri, for inviting me to this particular session today and requested me to give a lecture which will be beneficial for the students in the new syllabus. And basically, keeping in mind the benefits of the students, I have chosen this particular topic, that is techniques for estimating nucleic acids and determination of nucleolar frequency. For these two components, the nucleic acids and the nucleolus, most interestingly, both are residing within the nucleus, even though ribonucleic acid, one of the nucleic acid, it doesn't stay long in the nucleus and it is transported as ribonucleoprotein just after its transcription to the cytoplasm. Now, if we consider the estimation, any estimation that can be of two types, either that may be quantitative estimation or that can be qualitative estimation. And for the quantitative estimation, definitely it's the amount, and for the qualitative estimation, for these two nucleic acids, that is basically the purity. How much pure you get estimate that the nucleic acids, whether it is DNA or RNA. Now, if we consider, is it visible to all of you?
Yes, sir.
Sonali?
Yes, sir. দেখা যাচ্ছে।
Yes, sir.
Okay.
Audio, video, double clear.
And I am audible also.
Yes, sir. Audio, video, both clear.
Okay, thank you. Now, if we consider the DNA estimation, we can estimate DNA by different means. Like you can use the UV spectrophotometer for DNA estimation. We can use a particular staining, and I have considered this Dps staining as it is under the syllabus of the present CBCS, syllabus in the undergraduate course. We can estimate through Falcon microspectrophotometry, as well as we can qualify whether it is through the differential staining. Now, I will just give a short description on this. Through the UV spectrophotometer, what we do that the concentration of the DNA solution that is quantified by taking the absorbance at 260 nanometer, because that is the wavelength where the DNA is absorbed at the maximum level, both DNA and the RNA. And if we DNA, the formula for quantification is the DNA that is microgram per milliliter, that is equal to 50 into OD at the 260 nanometer of your sample into dilution factor. Now the 50 is as because the 50, the OD, when it is 1, that is the amount is 50 microgram per milliliter. Okay. And the purity of the DNA is obtained by the ratio of the A260 and A280. Now, in both the cases, DNA and RNA, you can measure these two, the amount in these two, the OD value in these two absorbance. And if the ratio is 1.8, that means you are getting the pure DNA. And if the ratio is 2, then you are getting the pure RNA. So while we are estimating both DNA and RNA, you'll have to be very careful to eliminate one of the nucleic acid to get the desired one. Okay. The second is through DPS staining, that is through diphenylamine staining. Now that is also you can quantify the genomic DNA through staining with this particular reagent, and you can measure it by taking the absorbance at 595 nanometer in a spectrophotometer or colorimeter. Now, difference in this between the two is that you require, in case of UV spectrophotometry, you require a spectrophotometer which is more costly because you need both the light source, UV as well as the visible light source. But if you go through the DPS staining, that particular method is very simple. It's very fast. It's not hazardous. Secondly, you need either a visible spectrophotometer or you need a colorimeter. So that means if you think about because I am considering about the undergraduate colleges where the fund is sometimes, it's a constraint factor. And that case, because the UV spectrophotometer, it may cost above 3 lakhs. I think so, maybe more than that, because I had one UV spectrophotometer in my lab, which cost probably 3.5 lakhs. And if you go for the normal this spectrophotometer or a colorimeter, it will be within 1.5 lakhs, not more than that. Okay, so these are the two techniques we can quantify quantitative estimation of that particular nucleic acids, both DNA as well as through the UV spectrophotometer, you can measure the RNA value also. Now, Falcon microscopy, that is very unique because the students, you must be knowing the C value. C value means the amount of DNA in the haploid genome. The 2C means the amount of DNA in the diploid genome. And 4C means when it is doubled. You can remember when the total amount of DNA is doubled, that is after S phase of the mitotic division. So after S phase of the prophase up to anaphase, this amount of DNA, it becomes 4C, and that can be estimated in situ, that is the nuclear DNA, through the microspectrophotometer by staining with Falcon or Scripps reagent. And the OD value, you can compare the OD value with a particular standard material like allium cepa, and you can measure the exact amount of DNA present per nucleus of that particular genome. This shows the genome size. Basically, through the in situ cytophotometry, we can determine the genome size of a particular plant genome. Okay. And lastly, the differential staining, that is the detection of DNA and RNA rich regions in plant cells using pyridine methylene staining. It's a particular double staining, what we call that. And through the double staining, you can definitely locate which region of the particular cell is DNA rich or which region of the particular region is the RNA rich region. And that is very interesting to see under the microscope. And I will just show you how it looks like later on. Okay. Now then, one by one we come. First is the estimation of DNA by diphenylamine reaction. What happens? The depurination of DNA, that means you can eliminate the purine factor, okay, that is from the DNA through either heat treatment or through the acid treatment. Acid hydrolysis, what we call that. And that 2-deoxyribose is released by the depurination of the DNA. And that deoxyribose undergoes dehydration, resulting in the formation of beta-hydroxylabule aldehyde. Now, this particular aldehyde, it condenses and reacts with diphenylamine and forms a variety of colored compounds, which together have a characteristic bluish color. That absorption maximum, it is at the 595 nanometer. That means in this particular experiment, we don't need the UV spectrophotometer because the 595 is a quite high, it's a visible range, within a visible range, okay? So that means the basic principle is that you have to do 2-deoxyribose that should be released, and that is released is usually carried out through the depurination of the DNA. And that deoxyribose undergoes the dehydration in the formation of beta-hydroxylabule aldehyde, and that aldehyde condenses and reacts with the diphenylamine that gives a characteristic bluish color. And through that color, we can measure how much DNA is present in that particular sample. Okay. What do we need for that? First, we need a standard DNA, and the standard DNA, everyone usually use that particular DNA, that is the capthaimazine or ctDNA, that is dissolved in 0.5 normal perchloric acid, and that is used as the standard DNA. Now, perchloric acid, it is basically that's a solvent where the DNA is dissolved very easily. And the genomic DNA that to be quantified, that is also dissolved in perchloric acid, which is 0.5 normal. So that means we need two different reagents. One is the perchloric acid, and that 0.5 normal perchloric acid is available commercially. You can have it from any chemical company like SRL or any other companies. The diphenylamine, that is a powder you can purchase. You'll have to purchase it from the companies, and then you'll have to prepare the solution. How it is prepared? 1.5 gram of DPA that is dissolved in 100 ml of glycolic acid, where the 1.5 ml of concentrated sulfuric acid that is added. So that means both glacial acid and sulfuric acid, they are needed, and the solution is mixed gently, kept in the dark in a refrigerator, and the DPA solution is prepared just before use, because you can't keep them for longer period. When you will do the quantification study, you'll have to prepare the DPA solution very fresh, and that's why, and you will have a good reading from that. How the procedure is? The procedure is that with the standard DNA, you prepare a series of dilutions. The standard DNA, the stock solution, that is 0.25 milligram per milliliter, that is in the 0.5 normal perchloric acid, and you prepare a series of dilution to give a concentration that is ranging from 50 to 500 microgram per ml. All the samples you'll have to prepare in triplicate. Usually, that is very safe to have it, and that is the very critical values that can be the errors that can be eliminated. Now, then you take 2 milliliter of each dilution of the standard DNA and unknown DNA solution, that is your sample solution, in 0.5 normal PCA, as well as the blank solution. What is that blank solution? That is the only 0.5 normal PCA solution. They are taken in test tubes, and to each test tube you add 4 ml of DPA solution. Mixtures that should be incubated in a water bath at 70 to 90 degrees Celsius for 15 minutes. So that means it's very simple. You take the DNA in different test tubes, and that 2 ml of DNA you'll have to take, and then you add 4 ml of DPA in each solution. You incubate the water bath, that is at the 70 to 90 degrees centigrade for 15 minutes. What we do, the tube 1 that is used as blank, and tubes 2 through 7 they are used for construction of a standard calibration curve for DNA, and the tubes 8 to 11 they are for the unknown samples. Now after incubation, the tubes are cooled down to room temperature under running tap water, and the absorbance values as well as optical density, that is as optical density, they are measured at 595. That means absorbance value which you are taking, that is actually as optical density, the OD value, they are measured at 595 nanometer against the blank in a spectrophotometer or in a colorimeter. A standard curve of absorbance at A595 versus quantity of the standard DNA that is constructed, and then the concentrations of unknown DNA solutions are calculated through extrapolation. Now, what we do basically, so this is the table by which you can keep this one, because this is the concentration of the DNA, that is the microgram. This is the, you are taking this amount of DNA solution, 200, 400 or like that, and this you can have this DHU that you add, that means it comes to the basically 2 ml in each case, and add 4 ml of DPA. Okay, and that is the way you do that. We do that. Now what we do in the standard curve, you get the OD values of the standard DNA at different concentration. Okay, and you put them on the graph paper. It's not that much clear, but I have tried to do that. You put the OD values for each concentration on the graph paper, and from the 0 point, you draw a straight line. You draw in such a way that maximum proximity of each data, the OD value that passes through that. That means if it passes through 3 or 4 OD values, it's very good, and the other OD values that remain very adhered to this particular straight line. And once it is done that, then you get the OD value of your sample, you put them on the standard curve, that straight line, and if you in the y-axis, that is in the microgram of the DNA, that is on the x-axis, you get that the how much concentration of DNA is in your sample. That means a standard curve is prepared by plotting the OD values of known concentrations of standard DNA solutions on a graph paper of A595 on y-axis. Okay, and that is on the y-axis, you get the OD values, and on the x-axis the amount of DNA that is in microgram per milliliter, that is you'll have to put that. So once you get that, you ultimately you'll be able to get the amount of DNA in your sample. And these are the cuvettes of the spectrophotometer, where you can have this, that is the blank solution, and these are the two, the concentration of this particular two, that is, it is much more higher, more blue than this one. That means here the concentration of the DNA definitely is less. Okay. Next, we are going to the RNA. That is another kind of distribution of RNA by Orcinol reaction. I have taken up this particular method as because that is also, it is under the syllabus of the CBCS course of the undergraduate course in the Botany honors, and that's why I have taken this by, and that is method also is very easy to quantify the RNA. That is the general reaction for pentosis, that is depurination of nucleic acid occurs after short exposure of RNA to boiling hydrochloric acid. And this extreme acidic condition catalyzes the dehydration of D-ribose sugar to form furfural compounds. So for the students, remember that in case of DNA, that is the deoxyribose, okay, and here that is the D-ribose, okay. That is the difference between the main difference between the DNA and RNA, and there the D-ribose sugar to form the furfural compounds, and these furfural compounds condense with the orcinol in presence of Fe3 plus ions of the ferric chloride acting as a catalyst and yields a variety of compounds to give green color with absorption maximum at 650 nanometer. So that means for the RNA estimation, we can use either a visible spectrophotometer or we can use a colorimeter. That means that is also convenient for the colleges where the fund constants are there. So that is the principle of this RNA reaction. That means the same thing, the defluorination, you'll have to do that through the hydrochloric acid treatment, and the D-ribose sugar that undergoes dehydration to form the furfural compounds, and these furfural compounds condense with the orcinol in presence of the ferric ion of the ferric chloride acting as a catalyst, yields a variety of compounds to give green color with absorption maximum at 650 nanometer. Okay. Now, what are the materials we need? The standard RNA that is dissolved in 0.1 normal sodium hydroxide solution, and the concentration that is like the DNA, it may be 0.25 milligram per milliliter. Now, reagent like DNA, that is the DPA reagent. Here we need the Orcinol reagent, and that's Orcinol reagent is prepared through 6% Orcinol that is prepared in dehydrated ethanol, and 3.5 ml of this solution is added to 100 ml of 10 normal hydrochloric acid, followed by addition of 100 milligram of ferric chloride, that is FeCl3. And the ferric chloride, as you know, as already told, that this acts as the catalyst, and the mixture is prepared like the DPA freshly before the use. And in both the cases, the longevity of that particular mixture is not that much, and you may get erroneous result if the Orcinol or the DPA mixture is prepared long ago. So you'll have to prepare maximum, you can keep them for 1 day, not more than that, even under the freezing condition. And that's why it's better to have prepared very fresh before, just before the experiment that should be carried out. Okay. What is the procedure? Like the DNA, in different test tubes, you take 2 ml of standard RNA in 0.1 normal NaOH of different concentrations, starting from 100 microgram to 500 microgram per milliliter, and another test tube, that is the blank 0.1 normal NaOH is taken and considered as the blank, and the unknown sample that is taken in a separate tube. You can have it is a triplicate, the unknown sample. Here in each test tube, 2 milliliter of the Orcinol reagent that is added, and all the tubes, including standard RNA and unknown samples, they are incubated in a water bath that is same at 70 to 90 degrees Celsius for 20 minutes. So if you compare these two techniques, the DPS staining for DNA and the Orcinol staining for RNA, they are almost similar. There are so many similarities except the reagents and except the study material. Otherwise, the technique is almost similar, and the principle is basically the similar, because you need to have the deoxyribose, in that case the DNA, and the D-ribose to release that particular from the chain, and then it undergoes the dehydration to form different compounds of different colors. Okay. Now, after incubation, the tubes are cooled down to room temperature under running tap water, and the absorbent values as optical density, they are measured at 650 nanometer against the blank in spectrophotometer. Similarly, a standard curve of absorbance, that is a absorbance 650 versus quantity of standard RNA, is constructed, and then the concentration of the unknown RNA sample solution is calculated through extrapolation. Now, here also you will have to measure when you get the amount. You'll have to think that that amount is the genomic DNA. Okay, that is the total amount. That is the genomic DNA or genomic RNA. Similarly, a standard curve is prepared by, that is a calculation, by plotting the OD values of known concentrations of standard RNA solution on a graph paper, and that is at the absorbance 650, okay, that is on y-axis, and microgram of RNA, that is on the x-axis. The amount of RNA in the unknown sample, that is determined by extrapolating the standard curve. Similarly, the same method, you get the OD values of different concentrations of the standard RNA, and you plot them against the amount of OD and the amount of RNA, and you get the different dots like here. You try to get the these are the different amount you are getting, and you'll have to, from the zero, you'll have to construct a straight line so that it passes maximally through the observance value. It may not pass all, like here also you can see that not all the points are passing. Maybe this point is not passing, this point is not passing, this is not passing. So that means as much as points that are OD values that are passing through that statement, that is good one. And once it is done, then you say you get the a particular sample solution where your OD value is here, okay, and you come straight down to that, and here the amount of RNA you are getting. So by putting your OD value on the standard curve of the unknown sample, and then that is the extrapolation of the standard curve, and then you get that, you get straight line to the down to here, and how much it is, you can calculate that particular amount of RNA. So that is the by extrapolation of the standard curve. Now the next slide is the different instruments what we get. That is the water bath. In the water bath, you may not you can have the water bath like you get a any utensil like what we use in the kitchen, in our kitchen, and you put them on the gas or on the heater, that can fill it with the water, and that can be when it is boiling, you just maintain the temperature in the boiling water and 70 to 90 degrees Celsius, and that is for the 20 minutes. Or if you are rich enough, you can have a water bath like this. And this is the UV spectrophotometer. Okay, this UV spectrophotometer, the middle one, this is the spectrophotometer. That is the UV spectrophotometer. Also, you can have the spectrophotometer without UV, only visible spectrophotometer. That is much more cheaper than the UV spectrophotometer. Or you can use the colorimeter like this. Okay. And for the in situ, nuclear DNA estimation, we use the cytophotometer. These are the two different angle cytophotometer, which myself and my students they have used this instrument for estimation of the 2C and 4C nuclear DNA to estimate the genome size of different plant species under my research group. And this is very old machine, and it was gifted by the British Council to our professor, Professor Arun Kumar Sharma, under whom I did my PhD research. And from then only, from 1981, I am using these two instruments for the DNA estimation. But that is very accurate result we get, but that is very old one. The present day cytophotometer is like this, the right one. This is very convenient to use it, because we don't have to have so many other accessories to get the actual wavelength of light to pass to the cell. But here, just by clicking the, it's a digital one, you put your wavelength on the digital box, and you get the similar wavelength for your study. Okay, so these are the two estimation group, that is the estimation of DNA and RNA. I hope the participants, they have I have been able to understand how the DNA estimation or RNA estimation can be carried out in the laboratory. Okay. Next topic is study of nucleolus using hematoxylin staining. I have taken this particular staining as because this is very easy and it is very fast. Okay. The nucleolus, the principle is the first of all, it's not an organ of a cell. It's very important. Like nucleus is an organ, mitochondria is a body, is an organ. Then chloroplast, that's an organ. The nucleolus, it's not at all an organ, or it's an object. Why? Because the nucleolus is the largest and most prominent membraneless domain in the interface nucleus. When a particular object has the membrane, you can call them an organ, because the nucleus has got a membrane, chloroplast has got a membrane, mitochondria has got a membrane. So everyone has got a membrane except the nucleolus, and that's a temporary structure. Basically, that is a temporary structure that appears during interface and disappears during late prophase. That means it doesn't stay long within the cell, within the nucleus. It appears and disappears depending on the time of the cell cycle. So when the cell cycle progression, through the cell cycle progression, it appears in the very interface, early interface, and it disappears during the late prophase. It is developed at the secondary constriction region, that is the NOR region. So that means the location of the nucleolus formation is very specific. It is developed in the NOR region due to continuous accumulation of ribosomal RNAs and ribonucleoproteins. So that is the region where the ribosomal cisterns are found in the chromosome. That means the NOR region is the region where the ribosomal cisterns are found, and from where the ribosomal RNA is transcribed. And for the continuous transcription of the ribosomal RNA, they are accumulated in the region, NOR region, and with the, usually you don't know, the RNA doesn't stay long as RNA. Immediately it converts into the RNPs or the ribonucleoproteins. And these ribonucleoproteins and RNAs, they are accumulated surrounding the NOR region and form the nucleolus. And when is the source of ribosomal RNAs and ribosomal RNPs, from where these are transported to the cytoplasm for ribosome biogenesis, and nucleolus associated bodies play an important role in transportation. Because here I have a specific experience on this particular topic. When I was in Laval, Quebec, Canada, I first worked on this particular problem, the nucleolus. Here, they observed a particular structure, and they thought that structure is probably the satellite portion of the chromosome. Because as you know, if you, when you study the nucleus, when you study the karyotype, you get the chromosomes with secondary constriction as well as primary constrictions, and you also get the maximum number of chromosomes with only primary constrictions. Okay, now these chromosomes with the secondary constrictions, they have a region that is very far apart from the actual chromosome, and that is called the satellite region. Okay, and this satellite region, they thought the group in that particular school of thought, they thought that that is the satellite of that chromosome, and that's why they invited me to clarify whether it is satellite or whether it is different thing. And for the first few months, I found that this can't be the satellite. Definitely, this is the particular thing where the nucleus associated bodies, these are called, and they are totally RNA rich bodies, and they are basically meant for the transportation of lump of RNPs and RNAs to the cytoplasm, as because I have traced that NABs from nucleus to the cytoplasm, nuclear membrane, and then afterward. So that's why this basically this nucleolus, and when the transportation of the RNA and RNPs occur at a very faster rate, the nucleolus, it starts to disappear very fast. When that means at a particular time when the cell enters into the prophase, it continuously transport the RNPs to the cytoplasm, and ultimately that's why it exists till the late prophase stage. The nucleoli start to appear in very early interphase at the NR region of the chromosome, and the number of nucleoli depends on the number of nucleolar chromosome. Nucleolar chromosome means the chromosomes having secondary construction. So that is, now you must be knowing that when we study the karyotype, we may not find the chromosomes with secondary construction under normal student's microscope. It doesn't mean that particular genome doesn't have any chromosome with secondary constructions or nucleolar chromosome. Each genome must have a pair of secondary constructions, because that is very important, and it contains the ribosomal systems, and you need to have the ribosomal RNA for the protein formation, because the ribosomal RNA, these are essentially required for the ribosome synthesis. And for the ribosome synthesis, as you know, it is the site of protein synthesis. So that means each genome should have at least a pair of chromosomes with the secondary constructions where the nucleolus will be formed. So that means it must be at least 2 or more or more per nuclear genome. In the late interphase, all the nucleoli fuse together and form a large prominent one. Okay, so if you study the nucleolar frequency, you may get the cell or the nucleus with 1, 2, 3, or 4 or more nucleoli, depending on the stage of that particular cell. The nucleolus that can be well fixed either in presence of formaldehyde or glutaraldehyde and stained with orcin, hematoxylin, pyronine methyl group, and the silver nitrate solutions. Okay. Now, the fact is that if you add aldehyde in the fixative solution, because usually when we study the chromosomes, we only use usually the acidic ethanol, that is acetic acid and ethanol, 1:3 or 1:2. But if we add the formaldehyde or glutaraldehyde, that is essential for fixation of subcellular organelles. That means if you use the formaldehyde or glutaraldehyde, you can fix the mitochondria, you can fix the the chloroplast, you can fix the nucleolus very prominently, and you can see them very prominently. The difference between the formaldehyde and glutaraldehyde is that if you fix with the formaldehyde, you'll have to use any one, either formaldehyde or glutaraldehyde. If you use formaldehyde, you can't quantify anything. This is only for the morphological purpose. But if you use glutaraldehyde, then you can have the quantify any component within that cell. Like by using the glutaraldehyde, you can determine whether through immunocytochemistry, whether the cell in the cell, which parts are protein rich, which parts are RNA rich, or which parts are DNA rich. But if you fix them with the formaldehyde, you can't quantify. So that is the difference between the formaldehyde and the glutaraldehyde in fixation. Okay. So that means if you start with the material, you can use the allium cepa, no problem. And the fixative, that is the usually it's called the FA, that is the formaldehyde or formalin, that is acetic acid, and absolute alcohol or ethanol, that is 10:5:85. Hydrolysis, either 45% acetic acid or 5-normal HCl. Any one of these two you can use. The fact is that With the hydrochloric acid, the cell is much more clear. But the problem is that there is one disadvantage. You may have the distorted organelles or the nucleolus. In the 45% acetic acid hydrolysis, you must get all the subcellular organelles very clearly, but probably it is not that much prominent to see. The stain preparation, the hematoxylin solution, that is the traditional staining mixture that is prepared by dissolving 1 gram of hematoxylin and 0.25 gram of iron alum. The iron alum is 100 ml of 45% acetic acid at room temperature. Very easy this is. And the mixture is homogenized with a glass stick and left in a dark flask for 1 week. After that period, the mixture is filtered and kept in the dark glass bottle. And you can use this mixture for long period. There is no problem. So that is the preparation of this hematoxylin solution is very easy. Okay, and you need only the hematoxylin and iron alum, because the 45 acetic acid is always available with the cytological laboratory. Procedure: the root tips of about 5 millimeter, they are fixed in a FA for overnight. Okay, and after washing in distilled water thoroughly, the tips are hydrolyzed either in 45% acetic acid at 85 degrees Celsius for 30 minutes in a water bath, or in 5 normal HCl for 20 minutes at room temperature. So depending on the hydrolyzing agent, you'll have to have 2 different types of method. If you use the 45% acetic acid, it is better have the 85 degrees Celsius for 30 minutes in a water bath. And if you use the hydrochloric acid, you can use it at the room temperature for 20 minutes, that is a 5 normal. And that will remove the basic proteins from the chromosomes and nucleus. After hydrolysis, the root tips are washed thoroughly in distilled water. Then they are further treated with aqueous saturated solution of iron alum for 10 to 12 minutes. The iron alum that is acts as a mordant to increase the stability, nothing else. You can prepare the iron alum solution separately, or you can mix them with the staining solution. But it's better to have the staining solution mixed with the iron alum. If not, you can prepare separately. And these are then directly transferred to 1% hematoxylin solution for 45 minutes at room temperature. Now, after staining, root tips are squashed in 45% acetic acid on a clean slide and observed under the microscope to study nucleolar frequency per nucleus. In the second method, after fixation and hydrolysis in 5 normal HCl, root tips are stained in a mixture of mordant and hematoxylin for around 20 to 25 minutes. Okay, and these are then washed and observed for investigation. So the method is very easy and it's not too lengthy. It's very fast. You can have it in a 3-hour class period. Now, for nucleolar frequency determination, the number of nucleolus per nucleoli, number of nucleolus per nucleoli per cell or the per nucleus, they are counted. So it may be you can have a single nucleolus in a cell, or you can have more than one nucleoli in a cell under 45x or the high power objective. At least 20 observations are taken to calculate the nucleolar frequency. This method of nucleolar staining is convenient as it is very fast. Also, double staining with the pyronine methyl group can be employed to study nucleolus, though it is term consuming. And this method can clearly differentiate DNA rich and the RNA rich regions within the cell. But that method is the double staining. Usually we did in our special practical class in our master's degree in Calcutta University, but now it is not done yet, as because we don't have that much of because the syllabus is quite high, and that's why we don't go for that critical staining methods. Okay, now this is the I can't remove this too, because the constriction is, you can see this particular picture where this is the nucleus, the white region, This is the nucleolus, okay, and you can see a clear knob-like structure. That is the satellite. It's a that is the paper I have written there, and that is the satellite region. And you can see there are reticulum like that, and ultimately that the two chromosomes they have come up. So that means we have studied a particular cell where only two chromosomes with secondary constriction regions are there, and they are close to each other, and ultimately they have formed that particular satellite. Okay, and here you can see that is the nucleolus. This is the the fibrillar zone, what we call, and that is the granular zone. And you see this one is the nab, because that is not attached with the nucleolus. You can see clearly it is separating out, and that is the nucleolus associated bodies, which is being it will moves towards the nuclear membrane for the the transportation of the RNAs and RNPs. They are also the nucleolus. This is the deep region, that is the fibrillar zone, and it is the granular zone, which is lighter zone. And this is also, you can see clear gap is there, because if you see the satellite, you don't see any gap, and they are connected, very much connected with the nucleolus. And here there is no connection between the nucleolus as well as the nucleolus associated body. And that's why in this particular paper we have clearly demonstrated, that is the first report, that definitely the nucleolus associated bodies are not DNA rich, and they are meant for the transportation of the RNA as RNPs. Now, when you go through the, okay, this is the hematoxylin staining method, and you can easily see that this is the early interface stage, where the number of nuclei are much more high. Okay, and if you go for the late, there is a single. That means the fusion of the nucleoli that occurs when by, and ultimately at the late interface, that is becomes at least one per nucleolus. Then that is the simplest microscope what we get in the college as well as in the university for the students only, poor students. Okay, this is one. This is the pyranine methyl green staining. Here you can see very clearly this picture. You see, this is the nucleolus. This is pure pyranine stain. You see the nucleus, there are greenish. I hope you can see that they are greenish. That is, they are methyl green stain. That is the region, the nucleus. That is the DNA rich region, and this is the cytoplasm. They are quite, they are pyranine stain. That is the RNA rich region. And here you can find that this is the two small bodies which are pink colored. That means they are RNA rich bodies. These are the NABs, okay, nucleus associated bodies. And if you see this one, this is quite green color, and that is the satellite region. So there is clear difference between the satellite and the NABs, and you can demonstrate it. We have demonstrated it very clearly with the light microscope only, and that is through the differential staining, the pyranine methyl staining. That is also, I think, you have in your syllabus. It's very interesting staining procedure. If it's possible, you can go through that after hematoxylin. Definitely I can help if you want that. And this particular staining, you can clearly differentiate between the DNA rich region, that is the nucleus, and the RNA rich region, particularly the nucleolus and the cytoplasm. Very very specifically you can detect these two regions, whether it is DNA rich or RNA rich. That is through the pyranine methyl staining. I think that's the all of today's lecture. I am ready to take questions if there is any. Thank you.
Thank you, sir. It was such a useful and more so needful explanation at the right time, I think, for all the important techniques of practical exercises that has been newly included in the undergraduate syllabus of University of Calcutta. And
Yeah.
more so, I would tell it because, sir, exams are nearly knocking the door. And
Yeah.
I think the concepts being clear about the principles of these various practicals is much more explanatory and much more vivid right now for the students, because they have not done it hands on. But it was so picture clear. I think there would be no, you have left no areas actually untouched for these kind of explanation. Thank you for making the practical so understandable also on the virtual level. But I think this lecture of yours will remain in our archives for a long time to come, sir, when we will be doing these practicals in the normal situation whenever soon it comes, and we will be in our practical classes, and your lecture will definitely help all of us. Thank you so much, sir.
Sonali, I'd like to
Yes, sir.
mention two things that
Yes, sir.
the first thing is that due to this pandemic situation, you know, I am I am always, I get interest to teach the students or to teach anyone who would like to know something from me.
Yes, sir.
I am very much interested with that. But due to pandemic situation, I am not familiar with this type of virtual meeting. And I really, with this today's experience, it's very pity for me that I don't see the expression of, you can remember, because when I usually give lectures in a university,
Yes,
I
sir.
used to see everyone's face, particularly the eyes, whether they are
Yes, sir.
getting interested with my lecture or not. And I used to say, if I don't, I see someone who is not getting interested, sometimes I used to say that, please, you can, if you'd like, if you can leave the class. But here,
Yes, sir.
I don't see anyone. And this is
That is the
really
con
very,
of this virtual teaching, sir. We can't help it right now because this is
and
the
this
new
is my
normal.
first experience with
Yes,
this.
sir.
Thank you for that.
But
And the
sir,
second
with a
thing,
turn up of more than 120 participants, I guess you would be really happy to know that you have reached so many people at the same time. I mean, all the students from various colleges have registered and have been listening to you, and it was such a wonderful experience, sir. Yes,
how
sir,
many?
carry on. So more than,
120.
yes, sir, more than 120. Yeah.
Oh my God.
Yes, sir. You have been catering to the needs of so many students at the same time. Thank you, sir.
Okay. And second thing is that, I am the alumnus of that particular college.
Yes, sir. We are proud of
And
that.
if the situation it becomes good, and
So
if
we would invite you to our department. Please, sir, do come.
if
We
I
would,
remain alive,
oh, sir, don't say that. Don't say that. You
I'll
are
be very
too,
happy to
you are
give
just
any
too,
lecture,
you are just
direct
too
lecture.
young to say all this, sir.
Ah.
Thank you, sir. I was just revisiting my classes during my MSc. My project was also with you. So maybe I have taken up this question answer session with you actually for that reason. I know you have students all over in my department also. My colleagues are all your students, but still I got this privilege to have this session with you. So before I carry on with the question answer session with sir's permission, I would just like to mention that the feedback link will be given in the chat box for all participants to kindly please do fill up the feedback. The time is given on your screens. If you can see, it's that feedback link will remain active till 11:30 p.m. So kindly fill it up to have your e-certificates within working days time. Thank you. So, sir, can we carry on with start with the question answer session? But before that, I would say you have cleared up all the queries, but still maybe there are some curious questions. There are a couple of questions I would really like to carry on with them. The first, sir, it came up like, what is the significance of nucleolar frequency, and is it compatible with the nuclear frequency determination? I mean, this came up like this only. What is the significance actually? Why would we want to know the frequency of nucleolus in a cell?
The significance means the Nucleolus, it's an object which is very important, in the cell cycle, as well as in the total functioning of the cell, because that is the region where the ribosome RNA is synthesized. And in that case, the nucleolar frequency also gives you an idea how many secondary constructions may be there. Because if you don't go through the chromosome preparation, at least through the interface cell, you can have that number. If you go through the interface cells, which are early interface, you can have an idea that how many chromosomes with nucleolar chromosomes or with secondary constructions they are there. That is the one of the most important significance of this study.
Okay. Okay, sir. Thank you, sir. Clear, sir. So next question is why it is particularly in situ nuclear DNA estimation. Is there any form of ex situ kind of estimation for DNA, nuclear DNA? It's just probably a question popped up. Yeah.
Nuclear
Srinjoy
DNA ex
Chattopadhyay.
situ formation is no, it's
No,
not there.
it's not
Because
there.
in
No
situ DNA estimation is essentially required because for the genome size measurement.
Yeah.
The genome size measurement is important because you need to know the genome size of a particular plant. If you go through the Arabidopsis, it's very small, where the 2C DNA value and 4C DNA value, they are much less than the others, like the alien.
Yes, sir.
Okay.
So another question.
There is no method for ex situ nuclear DNA.
Yeah, there is no method. So somebody asks like, usually DNA is the DNA PCA method is much more superior for the observation of the color change that happens, like because maybe some lady, I don't get her name. Let me just quickly. Yeah, Dr. Surendranath Halda. She has asked that they had probably done in their laboratory. They dissolved the DNA in the TE buffer, the trace buffer, and measured the OD. But DNA PCA method is superior for observational color change. Would you like to comment on that, sir?
Please repeat once more.
So Dr. Surendranath Halda, she has been doing this DNA. They dissolve the DNA in TE buffer and measured the OD. But she's mentioning that DNA PCA method is superior for the observational color change. Would you like to comment on that, sir? It's not actually a question.
When you dissolve the DNA in the TE buffer, with the DPS stain, you don't get any coloration. So the DPS staining, you must have the PCA.
Yes.
Okay, and that's PCA that gives the coloration ultimately.
Coloration.
Because in presence of the PCA, the deoxyribose that condenses with the different compounds, and that's why it gives a particular blue color.
Yes. So just a question. You mentioned.
Okay.
Yes, sir. So another one, like this is from our side. The DNA can be dissolved in sterile distilled water. Like, can we do that for this estimation part in the classrooms when we will?
Yeah.
Yeah, definitely. It can be done.
Not the DPS staining. No.
Oh, not for DPS.
You can dissolve DNA because DNA can be dissolved in normal distilled water.
Yes, sir.
DNA can be dissolved in TE buffer. But for the estimation, for the DPS staining method, you need to have the PCA.
Okay. Okay. So I think this would sum up the QA session because you have, I previously mentioned, you have like cleared all the principal concepts of the important practicals. I guess it's all clear now. For if there is any other queries, sir, we will pass it on to your email ID if somebody arises with some other questions. So thank you once again, sir, for making it so lucid and so so understandable for all of us. Thank you, sir.
Thank you.
So now I would ask Dr. Suchandra Chatterjee, coordinator DBT-Star College Strengthening Scheme, for as well. She is also our IQSC convener of our institution. I would like Dr. Chatterjee to please deliver the vote of thanks. Dr. Suchandra Chatterjee. Dr. Suchandra Chatterjee, are you? I would like our technical team to just go through whether she is present in our.
Okay, Sonali.
Yes, sir.
I think I found a question that is Srinjoy Chattopadhyay. Why it is called in situ?
Yes, sir.
In situ means you are not disturbing the organ. Okay.
Okay.
You are not disturbing, you are not taking out the DNA from the nucleus itself. DNA is within the nucleus and you are measuring in situ while it is within the cell. That's why it is in situ.
Okay, sir. I
Okay.
guess it
Am I
has a
audible, Shonali?
yes, ma'am. I was waiting. Yes, yes, ma'am. You are absolutely audible. Thank you, ma'am.
Oh. So, really warm spring evening, Bashanta Shandha to you all present here virtually for today's outreach webinar organized by Department of Botany, Surendranath College under DBT-Star College Strengthening Scheme. You know, the essence of all beautiful art is gratitude, and no duty is more urgent than that of returning thanks. And I feel myself really privileged and obviously thankful to Department of Botany for giving me this golden opportunity of delivering vote of thanks in this webinar. I missed their subject-specific webinar very recently for some unavoidable circumstances. But this time, you see, there is no miss. I always have faith in Department of Botany as one of the most promising departments of this college, so full of enthusiasm and creativity. Yes, the faculty and students alike, you are always so proactive, passionate, and responsible. I really love you from the deepest core of my heart and expect many more such type of programs in future to inspire all other fellow departments. On behalf of IQAC and DBT Star, I would like to
Ma'am, your voice has been disconnected, ma'am. Dr. Chatterjee.
She has to unmute herself, I guess.
and a really hearty vote of thanks to our honorable and much respected speaker today, who happens to be an alumnus of our college. Professor Mukherjee, we are so glad to have you among us today in this
Thank you.
outreach webinar. Sir, your most informative and useful deliberation on different techniques of estimating nucleic acids and on determination of nucleolar frequency will definitely enable our students to fully understand the subject. And as the topics are now in new inclusions in the new CBCS syllabus, the knowledge acquired from here is definitely going to help them to deal with the subject with sheer confidence in future. Thank you once again, sir, for coming here and delivering such an informative lecture in so eloquent manner, sparing your most valuable time. Today, though the meeting was virtual, but as you are an alumni, I earnestly request you, sir, to please guide and mentor your own department in future with your valuable advice and rich experience. Hope you will be with us for sure. Thank you so much, sir.
Thank you.
Now, I am immensely grateful to our college authority, the governing body, for their support, and obviously our principal, Dr. Indranil Kaur, for truly being an inspiration and standing always as a pillar of strength. Thanks are due to DBT, Department of Biotechnology, Government of India, for granting us this fund. We are really indebted to them. Special thanks are due to all the faculty, students, and laboratory staffs of Department of Botany for their tireless effort for arranging such a well-organized webinar, even amidst the ongoing university semester examinations. I thank all the dedicated members of technical team for helping in arranging such webinars one by one tirelessly. I am really indebted to them. Next, I must thank all our beloved students, both from our college and from other institutions too. Their spontaneous presence has no doubt made this event really colorful and meaningful. And last but not the least, valued viewers, all the dignitaries from different institutions. We owe you a lot for witnessing this webinar. Without your gracious presence, this event would not have been successful. So with these, now it's high time to conclude. And I conclude with a hope of meeting you all soon in similar programs in near future. Till then, goodbye. All stay safe, stay healthy. Thank you one and all. Thank you very much.
Thank you.
থ্যাংক ইউ স্যার। আমার মনে হয় মিটিংটা এন্ড হচ্ছে এখন। হ্যাঁ,
Okay.
I'll
তাহলে
call
শেষ
you.
করে আমি
হ্যাঁ, হ্যাঁ।